So the final speaker of the soil health session is Dr. Felicity Crotty. So I'm delighted to invite Felicity to speak next. Felicity joined the Royal Agricultural University as a lecturer in soil science and ecology in 2018. She is a soil ecologist working with the aim of promoting soil health and sustainable agriculture. She's particularly interested in how agricultural management impacts on soil quality, with a focus on biology, physics and chemistry. Through her expertise in all of these three areas, she's trying to disentangle the real impact that different management strategies can have on soil health and sustainability. Felicity has some current projects involving the use of AI to identify worm casts, which I hope we'll hear about this morning, and also linking soil biology to agricultural management and monetizing soil health. So really looking forward to your talk, Felicity, so over to you.
Thank you. Right, just sharing my screen, hopefully.
I see a cat.
That's good, always good. Okay, can you see that all right?
Yes, you just need to put it in presentation mode. Perfect.
Brilliant. Okay, it's not quite as obvious as it can be sometimes. Okay, well, thank you very much. I'm really, really honoured to be here amongst some very interesting speakers, and to also be talking about soil biology and soil health. So my original sort of starting point within soil science was soil biology and thinking about soil biodiversity, and that has led itself into thinking about soil health. So there's been quite a few different ways of talking about soil health. We've heard some of them this morning, in fact, thinking about the systems approach, thinking about looking at how all of the functions link together, looking at how you can use a soil scorecard with different indicators, and how useful potentially they are for agriculture. So however, within academia, there has started to be this push, you know, everyone is now publishing in soil quality, soil health, but there is a little bit of conflict. So starting to be this discussion of whether soil health is a useful concept, whether we should, where is it going to go in the future? Should we be discussing it at all? How does it work with the stakeholders? And actually, when you speak to farmers, as Elizabeth sort of hinted at earlier, is that actually farmers really do grasp the idea of soil health. And when you're starting to talk about something to the users, actually obviously it then is a very meaningful concept, and it's up to us as soil scientists to be able to start discussing it with them so that we can actually, you know, give the best advice possible. And when you're thinking about what makes a healthy soil, it does usually lend itself into an agricultural context, and it quite often it factors in soil productivity. But how you measure soil health is still much of a discussion. What do you consider to be healthy? Is it the ecosystem functions that the soil delivers? Is it the, you know, the nutrient cycling, the biodiversity, or is it just the provision of food and fuel? So a little while ago, sorry, I've lost my screen. Sorry, can you still see my slides?
Yes, we can, please do, thank you.
It's just me that can't. Okay, sorry. So a little while ago, I was doing, so I teach soil science to first years, and this coming year it'll be final years as well at the Royal Agricultural University. And one of the sessions I put in was looking at decomposition rates. And so I think probably a few of you have heard about the Soil Your Undies campaign, or the Bury Your Pants to look at decomposition. And thanks to COVID, actually, some of the pants that I buried did remain in the soil substantially longer than they were actually intended to. But it does give you this real obvious way of understanding what happens with decomposition. So this was buried at the field margin for the centre of the field for 100 days, and you can see that there's been a vast amount of decomposition occurring. This one was at the field margin, and it was for 100 days. So this obviously thanks to COVID. And it's just the seams and the waistband remaining. Some were ploughed in as well, I will add, and some I could never find again. You're starting to think about the function of the soil and how the biological activity is affecting these things. And when you start to think about whether you've got a healthy soil or not, you need to think about not just one soil function, not just productivity. It is about combining all of these things together. So, so we've seen sort of similar aspects to the little diagram on the right there a couple of times. And it's that link between the physical, the chemical, and the biological that makes up a healthy soil. But as my love is soil biodiversity, I do like to emphasize the point that when we're thinking about something being healthy, we are unconsciously acknowledging that it has to be alive. You do not have a healthy table or a healthy computer. You know, it has to be inherently living for it to be healthy. So we're thinking about the soil as this living ecosystem that isn't just an inert base for agriculture. However, the data that we have on soil health is actually very poor. We don't know within the UK how healthy our soils are, and we need to do more to investigate it. And it is these soil organisms that are driving the soil functions, the nutrient cycling, the decomposition. And agricultural practices will change this, will change the soil habitat structure and influence the abundance and diversity of the fauna within it. However, there is this huge abundance and diversity of fauna there. So within an agricultural soil, we've got probably a greater weight of soil fauna living below ground than you've got biodiversity, then you've got livestock above ground. So up to about 3 tons of soil fauna. So that equates to about a ton or a cow of bacteria. So a large amount of protozoa, mesofauna, nematodes. But earthworms are the ones I quite often emphasize the most, both because they're just a huge amount of biomass. So you've got calculated to be about 1.5 tons per hectare in your average arable soil, but it can be up to about 3 tons per hectare. So you've got this greater weight of earthworms below ground. They're really easy to see, to count, to understand what they're doing. And they're also these ecosystem engineers. So if you've got a healthy earthworm population, then you have then the likelihood is that the other soil fauna are also quite healthy. So so these are the ones that are driving water infiltration, mixing the soil up, changing the where the nutrients are within the soil profile as well. So they're helping the other organisms as well. And it's been found in a number of studies that they can actually increase crop yield. So if you compared a field with 0 earthworms to a field with a healthy number of earthworms and in a low nitrogen environment, potentially you'll have this 25% increase in crop yield. So knowing how many you have could be this quick and easy gauge of soil health. I'm not advocating drilling down to one indicator, but if you have a healthy population of earthworms, then the chances are that there will be a lot of other components that will also be healthy. And the benchmark for your sort of not in a sandy soil context, but in a sort of clay soil context, your benchmark would be about 16 per spadeful, which equates to about 400 per metre square. And in the UK, we've got a number of different functional groups. So we've got the epigeics, the surface dwellers, the dark black worms that live near the soil surface, eat recently fallen organic matter. They reproduce quite rapidly, are quite short-lived, but are quite disturbed by agricultural management, so tillage, for example. Then you've got the soil dwellers, the endogeics. These ones are horizontal burrowers. They live in the soil profile and they just eat soil, so they're very rarely at the soil surface. And these ones are sort of medium-sized worms, quite pale, reproduce fairly rapidly, and live sort of a medium length of time. And these are the ones that you probably if you do a soil survey, about 70% of your worms will be the endogeics. And then you've got the anesics, the big vertical burrowers, Lumbricus terrestris, the ones that move up and down the soil profile, that eat, that take organic material from the surface and they pull it down into the burrows where it then decomposes, and then they consume it there. So they're redistributing this organic material, they're creating permanent burrows, which allows water infiltration. However, they're very slow to reproduce. So it can be that agricultural management affects this group the most. Now in the UK, we've got about 30 native species, so not actually a huge amount of diversity for earthworms, and that's what makes them an ideal indicator, particularly in the UK, because these are the ones that you can pretty much commonly find, easy to identify as well. It's unlike springtails and mites, which I did for my PhD. 30 species, not a problem. 200 species of springtails and mites, bit of a problem. And also you can see them really easily with the naked eye as well. So I was recently involved as part of a huge global analysis of earthworm distribution, and what they found, which again links into this issue with global change, is that earthworms are more abundant and diverse in temperate regions than at the tropics, which is the opposite to what happens with above ground biodiversity. So if you think about a warming climate and how important earthworms are within agriculture and within a healthy soil, there is this chance that we're going to be reducing their numbers and could lead to large problems in the future. So knowing earthworm numbers is key to this, and I'm just going to go through a couple of different experiments where we've looked at earthworm numbers and how that relates to healthy soils. So first of all, there was an experiment at RAU, it's a bit of a demonstration experiment where we discuss looking at tillage versus no-till. And it's historic. It's historic that we all know that tillage, ploughing is bad for earthworms. And the thing about this is that what we don't really discuss is that it's fivefold. So if you think about what happens when we plough, you've got the direct damage to earthworms, the plough cutting up their large bodies. Then you've got the physical disturbance to the soil habitat, so cutting up the permanent burrows of the earthworms. We're burying the crop residues, which means that they're then having to expend more energy to try to find food. And then if they've tipped the world upside down and they're now on the surface, they're at a greater risk of predation as well. And then at the surface, you've got this more extremes in temperature and moisture as well. So the more chance of them drying out or being affected by the higher temperatures than they would be if they were buried within the soil. So all of that leads to a reduction in earthworm numbers. So in an experiment, it's part of a demonstration for 5 years, was looking at different methods of crop establishment. Now we all know, as I say, that ploughing is bad, but what about no-till? And are all no-tillage methods equal? So in this case, we were looking at 3 different direct drills, the Missouri, Seedhawk and Crosslot. And when you look at publications, it's actually really difficult to find, it takes a very focused amount of reading to find out which direct drill or which no-till was used within the literature, which is something I've been, this paper's in preparation at the moment, but you'll see that it's less obvious than when you say ploughing. So looking at the effect of these different drills, there were differences found for earthworm numbers. Now they were all quite low, and I'm putting this down to we had 2 years of relative drought. So 2018 and 2019 were pretty dry, sort of April to June, which is when earthworms are quite active. They're less active over the summer, which would be sort of June, July, August, but actually when they're usually more active, the soil dried out. If soil dries out, they don't do a lot of moving around, and so then less foraging, less fit to survive over the winter period as well. Now, what was key with the results in this experiment was that the seed hawk actually had one of the lowest measurements for epigeics and anisic species in comparison to the Missourian crosslot. Plough didn't have any epigeics at all, was found, and suggests that, as I say, these differences in tillage system do affect earthworm numbers. Not all tills are, or not all no-tills are equal. Same was true for compaction within these plots. There were changes within that. So you can see that each drill was affecting the soil structure to a varying extent, particularly at the surface, and then as we get down to 20 centimeters depth as well. So differences in crop establishment between these drills, and that we need to consider this when we're talking about tillage versus no tillage. Now, how we measure earthworms is quite time consuming. If anyone's ever done it, you take a block of soil, you break it apart, you pull out as many earthworms as you can find. Does create quite a large bit of soil disturbance. So if you're doing it in a farmer's field, I mean, you know, you've got hundreds of hectares to go on, so it doesn't matter so much. However, if you're doing it in a site of special scientific interest or a nature reserve, then they might not be so keen. It also takes a little bit of taxonomic expertise. So I started off by saying we've only got 30 species. It's really easy to identify them. And I always used to say that until I started teaching my first years how to identify earthworms. And then I realized that the majority of them, particularly virtually, they were better when we did it face to face, actually kind of all put them in as compost worms or brandling worms. And you don't actually find them very often in an agricultural field. So my suspicion over their identification skills was that they weren't particularly accurate was quite high. So they're pretty good at identifying adults to juveniles, and I think quite a lot of us are. You can see the saddle, but actually identifying earthworms to species by non-experts is not as easy as I first thought. So what should we do to get more people counting and identifying earthworms? Well, one of the other methods I've been recently looking at is counting earthworm casts. I'm wondering if this is a more reliable method and whether we could develop technology to do this for us. So I'll just thank my collaborators now because I'll forget later. So this is a project sponsored by the Science and Technology Facilities Council, looking at whether we can use computer algorithms to develop earthworm identification skills. So whether we can start to estimate earthworm activity within agricultural fields without having to rely on experts, and whether that means that we can cover much wider areas as well. So this is quite an early stage in the project, but what we've been doing is setting up sort of quadrats, putting out photographs of the casts. Sometimes we've been ringing them, or we've got a different setup here. So we can create this library for image detection and recognition by the computer. And so these are zoomed in pictures. So these are about 20 centimeters squares. What you can see is you're starting to see a lot easier to see the earthworm casts on the soil surface. However, brown on brown is quite difficult. So looking at using TensorFlow 2 model, training the models to be able to identify the earthworm casts. So we need lots of pictures, and what we're doing is basically we're drawing bounding boxes around the casts, so that then the computer system can go through them and be able to tell what is a cast and what isn't. However, as I say, thousands of images are needed. And so each of the bounding boxes contains the coordinates of where the object resides in the image. So you can see that we've got picture on the right there. You can see it's managed to find a lot of the casts. However, there are a few that I can see haven't been drawn around. So it's about working out whether we're getting some false positives or false negatives, and getting the computer system to be as good as a human is in the field, but also so that we could then take it out and get other people to to take the pictures and being able to calculate whether they're finding all of the casts. However, at the moment, it's been very dry and we've had very few casts on the soil surface. And you can really see the difference in earthworm activity over the course of the year when you're trying to go out in between lectures and take pictures of them. So just to conclude, I think I've actually been relatively good time there. Using something like earthworm casts will help us to monitor earthworm activity across the landscape, and being able to, the soil is so heterogeneous that we can't, so just taking one spot in a field, or even like one small area within a field, and being able to extrapolate that for the whole field is unlikely to be a useful measure, because we all know that there are some areas which are more compacted or more waterlogged than others. So actually looking at how it varies will be really useful, and doesn't often happen within monitoring terms. And as we all know, a healthier soil is there'll be more functional activity, and it'll be better in the long run for both the agricultural productivity, but also all of the ecosystem services that the soil does provide. And so I'll just conclude there. So hopefully, if you've got any questions, I'd love to have them. Thank you.
Thank you, Felicity. That was fascinating. Image detection of earthworm casts, that's absolutely brilliant. I definitely want to speak to you about that sometime. Really, really good. So we're going to go into questions now. So I think Dan's been monitoring those. Once we've had a few questions for you, we're going to go straight into our panel session. And for those of you who are desperate at this point for a cup of tea or a coffee, we're going to have that as soon as we've finished our panel discussion. So please just hold on for a moment, and I'm going to now hand over to Dan.
Thank you, Sasha, and wonderful talk, Felicity. So we got a couple of questions here, and I see there are some trickling in. So I'll try to keep an eye on those for the next 5 or so minutes, and then we'll bring the panelists together, as Sasha said. So the first question here, Felicity, is what about DNA for earthworm species identification? I guess it's a different method, an alternative method. Have you got any thoughts on DNA?
I mean, we can use DNA for earthworms. The question is, what are you measuring? How much field do you want to measure? So you could take a small, for bacteria, you take a gram of soil potentially from a whole field, and you identify the DNA within it, and you understand what microbes are there. The same you could do for earthworms. You just need the different primers. However, was the earthworm there? Is it the earthworm cast? Is it the earthworm poo you're detecting? Is it skin cells that are sloshed off the earthworm? Were they there now? How long does the DNA last within the soil? It ends up opening a can of worms, potentially, with more questions than answers.
Yeah, pun definitely intended there, I think, with a can of worms. So this is an interesting one then. So could you argue that with climate change, while earthworm distribution may change as temperate regions warm, the overall ecosystem services that these worms provide will still be provided, because there'll be an upward migration of the tropical equivalents, such as ants and termites? You got any thoughts on that, Felicity?
I suppose it depends on where you are within the globe. So I think it would be relatively difficult for the UK to get the tropical equivalents of ants and termites moving into our area. I mean, potentially. I think the main issue with climate change is that these sort of occurrences aren't going to happen quick enough. So more the detrimental effects are going to happen much quicker than any of the positive effects of migrational movement of species. So you're looking at potentially a period where you're going to lose the function before something else can fill that niche. Now, chances are that in nature that a niche gap doesn't usually stay a gap for long, so something will try to fill it, but it will take time. So it all depends on, are we talking about tens of years, or are we talking about hundreds or millennia?
I guess that's the question for the panel as well, as we talk about scales as well. in soil science. So